Now we're going to talk about the specific case of vision, literally how you see things. And for this, it's a process that's going to involve a G protein coupled receptor. And the name of this receptor is going to be one of these things. It's either going to be rhodopsin, which is the one you get in your rod cells. So think of it as rhodopsin, but actually it's rhodopsin. And then you're going to have your three opsins that you find in the cone. And so you can call those photopsins. And these are the ones that are going to help you see a little bit better in the green, red, and blue. And rhodopsin is basically just going to show you light versus dark in general, or rather that's how we're going to use it. And each of these is a G protein coupled receptor. So rhodopsin and the photopsins, the three photopsins, are all G protein coupled receptors. And each cone cell is going to make one of the photopsins, so it's going to be a red, green, or blue cone, or it can be a rod cell and it can make rhodopsin, in which case, it'll just see generalized light, or that's how we will interpret it. So the question of how does a protein actually sense light? This is a very interesting one, I think. And you could go certainly a lot further into the physics. There is some very interesting physics here, but we're going to stick a little bit to the surface. We'll go a little bit into it, but not too far. Just at least to the level where I think you can see kind of how it works. And this is going to be one of the test questions for sure for the second exam is, tell me about the function of GPCRs in vision. And this is it. So, all right, let's talk about these. All right, so each of these opsin genes, you see opsin is the part that's in common, the photopsins and rhodopsin. So you can call them all opsins, that's fine. And that's the ancestral protein that they're related to. And some animals that can see light but are more primitive would only have a general opsin gene, for example. Each of these is going to hold on to a special molecule called, right here, I'm circling it, 11-cis-retinal. And it's like retinol, but with an A at the end, retinal, because it's not an alcohol. So let's look at this thing, and this is showing you the different wavelength, where each of our opsins binds best. So 530 is pretty close to the center of green, and then you got your blue and your sort of yellowish green, which we're going to interpret that plus rhodopsin as red signal, I believe. All right, here we go. Okay, so as it turns out, molecules can absorb light. So just like we talked about in photosynthesis, the light is going to come from the sun or a light bulb or whatever in the form of these little tiny, not quite wave, not quite particles. They're just little packets of energy, and they are called photons. And what can happen is, so this is just an example. This is not 11-cis-retinal. I'll show you that in a minute. But this is showing you a generalized case for what can happen. So you can start with a trans molecule, and by trans we mean that in this case it's kind of going to have a zigzag shape. Is basically how you can think of it. So the two big groups are going to end up on opposite sides like this. It's just going to zigzag back and forth. The cis conformation is actually all the same atoms. We've just rotated, so we've twisted this bit around. Rotated one particular bond, and so now, boom, we're locked down. And there's some rotation that is allowed in molecules if they have a certain amount of energy. They can have a little vibrational energy or something that makes it a little easier to rotate from one to the other. But generally, once it gets into the trans conformation or the cis conformation meaning shape, and here's the word conformation right up here. When it's in either of those, it's kind of stuck in an energy well. So here's our energy well down here. So here we're in the cis energy well, and we're kind of stuck. We could get a little energy, and we just slide up here, and then boom, slide right back down. Get a little energy this way, slide up, slide right back down. Unless we can get enough energy to bump us over the hill and into here, or enough energy to bump us up the hill into who knows what lies on this side. Same thing for the trans down here. And so what we're showing at the top is the amount of time. So this is the transformation. And it's actually going to go through a transition state. That's our TS right here. And so there's going to be a little confirmation that's somewhere in between this and this. And I think if I was better at chemistry, I could tell you exactly what we think this would look like. But those transition states are very, very fast. Yeah. Here we're talking about in the order of picoseconds for these transitions to happen. And so our transition state, we're going to go down into this energy well for about 70 picoseconds and then up and over the hill in about 3 picoseconds to get to this sort of twisted species in the middle, which is highest energy, but which is able to then just resolve to this because we're using the energy to go up the hill here, and then we come back down the hill here and we land in the cis conformation. Yeah, basically it's just a case where you put in enough energy and a particular thing will happen. A molecule will change shape. So let's make this bigger and let's turn our subtitles on because that makes it nicer. There we go. So now we'll have some text on the bottom. That'll help, won't it? Okay, so this is the actual molecule that we are talking about. So we've drawn it out here as if it has all of its atoms. We're not scrimping on any of those atoms. And so we got a ring at one end with a couple little branches coming off of it. And then a long snaky molecule that comes down here. And what we have here is 11 cis retinal. So you can see on this side we have this hydrogen and this hydrogen that are on the same side. Cis means same. And so when you have hydrogens that are next to each other that are on the same side, then that is going to be a cis version of this, conformation rather. And if we were to rotate this entire molecule 180 degrees, then without breaking this bond, we can just sort of twist around this bond. Like imagine that one half of the molecule is your hand with your index finger pointed out, and imagine you got your other hand there, and then boom. That thing where you're touching your two index fingers together, that's the bond. And notice how you can rotate both hands, and it's fine. The bond can remain intact, but yeah, you've rotated around it. And so once again, for these molecules, when it's in the cis conformation, it's in a fairly stable spot, but it could get to a slightly lower energy conformation, which is the all trans conformation over here. And this one is all trans except for the 11th carbon is cis. So that's why we call this 11 cis retinal, as you can see up here, 11 cis retinal. Okay, and so this thing is kind of locked in. So there are little interactions between these molecules on the side, and there's basically not enough energy in a just normal molecule for it to actually twist around that bond. But when you put in a photon, it gets just enough energy, and now you get efficient switching, efficient rotation around that bond, and then boom, you're going to end up in the all-trans shape. Notice the difference between these two. All cis is kind of bent. You see that? It's got like a 45-degree angle in it. And all trans is like a long, straight rod. Now imagine what would happen if you had a protein that was locked around 11 cis. And imagine it stretched out. It's just like if you were sitting in a cardboard box and then decided to stretch out your legs. Boom, you change the shape of the box and or destroy it. That's the way that your proteins are going to see. The photon will come in, and the protein is going to be loaded with this 11 cis retinal molecule. The protein is actually going to act like a lens, and it's going to focus the molecule right onto this little part right here. And when we've got enough energy that happens to have landed there, we've got enough photons delivering energy, then we're going to be able to switch. And we're going to switch over to the all-trans conformation. That is going to change the shape of the protein, and the protein, which is a G protein coupled receptor, is going to kick the G protein, and the G protein will go off and yeah, do its thing. Exchange GDP for GTP and go on and activate something. All right, so here is a protein, and here is the same protein when it is activated. So this one is rhodopsin, the opsin that you find in rods, and we've got the inactive form in green, and we've got the active form in this sort of, I don't know, kind of not quite coppery color, whatever, metallic brown. Yeah, not quite platinum either. I don't know what you call that color. Yeah, yeah. Anyway, so you can see that on these parts, the alpha helices pretty much line up. But right over here, helix 5 and 6 are actually going to get pushed way out to the side. So they were kind of tucked in around the center of the molecule. And when our retinal, so this bit up here is like the lens. This is going to focus the light down onto our retinal molecule. So all the light's going to come in and boom, smack right into that. It's going to change shape. And so once it's stretched out nice and long, it's going to push out this part of the molecule. Just like if you were in a cardboard box and stretched your legs out, you'd push out one of the walls of the box. And so helix 5 and helix 6 are going to move outward by, it says by about 7.7 angstroms. And an angstrom is about the size of a hydrogen atom, something like that. And it's roughly the size of an intramolecular bond. We're in the right neighborhood anyway. Okay, so here's our change at the top. We're going from 11 cis retinal plus a photon, and we're going to transition. It's going to be an isomerization. We're just going to rotate this thing, and we're going to turn into all trans retinal. And when we do that, we're going to push out the side of the molecule over here, helix 5 and 6 in particular are going to move. Okay, and so that's what it's saying at the side. So this is these are the first steps of the answer. We're kind of pinched in around the 11 cis retinal, then when the light hits the 11 cis retinal, it is going to straighten out into all trans retinal, and that's going to force, in particular, helix 5 outward. That's the one that moves the most. We'll move a little bit of helix 8 as well, and helix 6 is going to move also with that. There you can see 5 is really getting pushed quite out of shape. All right. So, yeah, rhodopsin is part of this giant system. So it's one of many proteins that make vision possible, but they're all doing different parts of the task. And rhodopsin is actually the one that is most responsible for carrying out the light binding and starting this off. So that's what we've got over here. We've got our retinal. See the little ret right here? And so which retinal molecule is this? I see there's our ring at the end. There it goes. And then it's bending off at an angle, which must mean this is 11 cis retinal, right? Yeah, of course. And then when we absorb a photon, so remember this bit up here is going to act like a lens to focus all the light right down in here on this little bond right at the center of the retinal. It's positioned right about at the center of this little barrel. And then once that happens, we're going to stretch out, and the back half of the molecule basically stays there. The front half with the ring on it is actually what's going to push out. And we're really going to push on helix 5 right here. And so helix 5 is going to get pushed out. Helix 6 is attached to it. That's going to get pushed out a little bit. And that's going to be the activation. So that's just showing it in another way. All right. The rhodopsin itself, so we're going to end up with this form down here, the all-trans retinal. And then it turns out there's a process, and there are a series of enzymes that will reset this molecule and turn it back into regular old 11 cis retinal. And so we've got this thing where it's going to pop out of the rhodopsin. We're going to load a new 11 cis retinal into the rhodopsin. And then once it is in the rhodopsin, it's going to absorb light, and then bam, we call this photorhodopsin or all trans retinal. There it is, all trans retinal in the world's tiniest writing. And then it's going to go through some other minor changes. And then we're going to send it off, and we're going to recycle it and turn it back into 11 cis retinal. And so the body's going to actually have to use energy in the form of probably ATP or something like that to reset this molecule. But it's fine because it allows us to see. So vision is going to burn energy directly, but we're not actually burning energy in the process of receiving the light. We're just putting the right molecule in the right position to succeed. Isn't that nice? All right. The bottom thing shows basically how the signaling works after this. We're going to look at a larger version of this, and I think that's going to make it a little bit clearer. Yeah, that's my hope. So yeah, rhodopsin and the other photopsins make vision possible. They all basically work the same way, so just focus on rhodopsin. That's going to be good enough. And the other photopsins do the same thing, but in a different kind of cell. And they're going to be more activated by different wavelengths of light. And so big conformational changes, particularly to helix 5. And there's a little thing down here at the bottom that they sometimes call helix 8. That's also going to move a lot. That's the part that you saw go from out like this to sort of twisted up like that in the first video that we watched. So in the other one that I've recorded for you. And yeah, the change in helix 8 is the one that's actually going to affect the G protein itself. So the G protein is going to be bound to GDP, and it's going to get rid of that GDP, and it's going to bring in GTP. And once it does that, it's going to be active. The alpha subunit will split off from the beta and gamma. And there we go. It's going to activate a thing called a cyclic phosphodiesterase. And so that's PDE. And here's the word, cyclic phosphodiesterase. And yeah, it's going to make a thing called cyclic GMP into normal GMP. So let's look at what that means. All right. So this is GMP, guanosine monophosphate. If we wanted to turn this into GTP, the nucleotide, we'd have to add 2 more phosphates. So we put 1 more. So this is a phosphate, P and the 4 O's right here with a negative charge. We could put another phosphate on here and another phosphate attached to that one. Then we'd have 1, 2, 3, and we'd have GTP. But it only has 1, it's GMP. So GMP is good for a couple different things. One is that we can charge it back up and make it into GTP, which is great. Another thing we can do, though, is we can twist it around and make it into cyclic GMP. And so this is cyclic GMP over here. And it's got an extra little ring. That's the cycle part in here. We've made a little ring. And you can see it's just one of these atoms from the phosphate. So we're going to lose this hydrogen, and we're going to have 2 oxygen with a negative charge. Oxygen with a negative charge is going to attack at this carbon right here. It's going to push off this OH hydroxyl group and this H+, and together they're going to make a water molecule. And so that water molecule will go away, and we'll end up with this oxygen joined on right there, which is going to make another little ring next to this is the ribose ring. And if it was deoxyribose, it would be missing this hydroxyl here. This is the first position, this is the second position, third, fourth, and fifth. And so deoxyribose is 2', second carbon deoxy, and so it's just missing this oxygen. And it just has the hydrogen instead. All right, so this one I like a little better for showing what's going on. So in the first half, we had our rhodopsin protein that had the 11-cis retinal, and it absorbed 1 photon of light. And once it did that, the 11-cis retinal changed to all-trans retinal. And now that we've got all-trans retinal, it's going to change the shape of the retinol molecule. Or rhodopsin molecule, sorry. And the rhodopsin molecule that's bound the light here is now activated. So we're assuming light's coming in, and then this is R star, and R star means R activated, rhodopsin activated with a photon in there. And so this is going to change shape in such a way that it pops out this GDP. So we're going to kick out GDP, replace it with GTP. And so now our alpha subunit, this is our G protein here, alpha, and this is beta and gamma together. The alpha subunit is going to go off and do things. And forget about the beta and gamma, just imagine they go off and do something, which in this case is not quite as important for our test answer anyway. The GTP containing alpha subunit of the G protein is then going to go over here to this thing, which is the phosphodiesterase. And phosphodiesterase takes cyclic GMP that's kind of bound up into a circle, and it's going to turn it into regular old GMP. So cyclic GMP is a neat molecule. These cyclic nucleotides are used in signaling, and for example, not that you ever have, but if you would ever take something like Viagra, this is another molecule that is going to affect the production of cyclic nucleotides. I think it's cyclic GMP in that one as well. And that's how it works. That's how it does the thing that it does. Yeah. So kind of neat, right? All right. So our GTP containing G alpha subunit is going to go over here and it's going to activate the cyclic phosphodiesterase. So the phosphodiesterase has alpha, beta, and 2 gamma subunits. The gamma subunit is the one that's kind of blocking the alpha and beta from doing their job. And so the activated G alpha subunit just pulls the gamma subunit of phosphodiesterase away. And so now phosphodiesterase is going to be active. And so if there's any cyclic GMP around, it's going to take it and turn it into GMP. And you say, good job. Way to ruin a nice cyclic GMP molecule, but why do we care? The answer is because this cyclic GMP is like a key. So cyclic GMP is a key. And when you have cyclic GMP, there are channels, actual ion channels that are going to be open that are going to allow calcium and sodium to come in. So we're basically going to keep the cell kind of positive as long as we keep that channel open and as long as we have cyclic GMP. So yeah, cyclic GMP, the little C, think of it as like a little smile. It keeps the cell positive by keeping the channel open. You can also think of it as a channel key. That could be another reason why it's got a little C there, even though it's for cyclic. Okay, and so the reason why we need to keep bringing in positive ions is because we have this exchanger, which is going to constantly be bringing in a little bit of sodium and pushing out calcium. And calcium is going to have a, this should just be Na+, not Na2+. But calcium is going to have a double positive charge. And so this one is always going to be pushing out positive charges. And the other one is always going to be letting in positive charges. And so, yeah, positive charge comes in through this and goes out through that. And as long as they're both working, then the cell's going to stay neutral. But if we close this in gate, so we don't let any more positive in, then we're going to build up negative charge inside the cell. And when we got that negative charge inside the cell's going to go negative, and it's then going to fire off. Because the rod and cone cells are able to send an electrical signal to the neurons that are attached to them. And that's the next step. That's how we're actually going to see things. So we're going to focus on just what happens inside of the rod or cone cell. That's the process that we're trying to talk about here with the rhodopsin and the G protein coupled receptor, which is rhodopsin, the G protein, which gets GTP, the cyclic phosphodiesterase, which breaks down the cyclic GMP, and the little channels that were only being kept open by cyclic GMP. The cyclic GMP actually it'll bind to these for a minute and then it'll fall out for a minute. And so it keeps them open, but not for long. And as long as there's a lot of cyclic GMP around, then it's fine. They will stay open. But as soon as we start breaking that cyclic GMP down, it's going to close. And then the cell is going to get real negative, and then it is going to signal to a neuron, which is going to carry it to your brain. Now remember that all this has to happen fast enough, because if somebody throws a baseball at your head, or hits a baseball at your head, let's say, we have to do all these steps fast enough that you can fire off that neuron to the brain, interpret the signal, and send some kind of signal back, to the face or the hands or something that says duck or get out of the way, or get your glove on that fly ball, or else it's going to hit you square in the face. And so thankfully this works really fast. And I think your challenge is going to be trying to be able to go through the process enough that you can get it really quickly down so that you can talk about it. And so here is what's going on with the ion channel. So the ion channel looks roughly like this. When it has cyclic GMP, it's going to bind right here, and it actually sort of pulls the channel apart. So this is the channel when it's closed, and when it's got cyclic GMP bound, it just sort of pulls the drapes back, if you like. which is going to allow these ions to flow through in one particular direction. And then with the cyclic GMP pops out, if there's not another one to go back in, then the drapes close and we're back to this situation that you've got over at this side. So there we go. This is exactly what happens. Yeah. The vision, just imagine it's looking at bad things and it's just trying to stay positive. You got to stay positive. But when we close that gate, when we take away the cyclic GMP, then the cell's going to get negative, and when it goes negative, it's going to fire off a message. Yeah, just like intracellular Twitter, and yeah, then stuff's going to happen. There we go. So that's the end of the lecture. I'll go record another version where I draw this out on the whiteboard when I have access to that tomorrow. So look forward to that, and thank you very much.